In this video, we're going to talk about antibody anatomy and some of the basics of antibodies. We'll cover this again in chapter 15. But antibodies are basically they're Y-shaped proteins. Okay, so they all have the basic overall shape. They have a what's called an Fc stem, and on the tips of the Y would be a variable region that would interact with an antigen in a specific manner. The antibodies are part of the adaptive immune system, so this variable region interacts specifically to a antigen shape. So again, an antigen can be a virus, a bacteria, fungi, things like that. Um, very much in a lock and key type theory. So that variable region would recognize something specific on the antigen and attach to it. Antibodies are produced by activated B cells. And once they're activated, they're called plasma cells. So plasma cells will secrete antibodies during an adaptive immune response. So it takes about 2 weeks for the highest level of antibody to build up in the body during a primary immune response. One of the nice things about the adaptive immune response is that we get memory cells. So memory B cells would be involved in a secondary response, and we have antibody produced 3 to 4 days. So you might get a mild infection, but the antibodies would clear that up relatively quickly. Now, like I said, the antibodies are going to attach to an antigen in a specific manner. So if we have a bacteria, there would be what are called antigenic determinants on the bacteria cell surface. So it might be the adhesin pili, for example. That might be the flagella, different things like that. So these antibodies, variable region, would interact specifically with an antigenic determinant on the actual antigen. Once you have this antibody attached to the antigen, the Fc stem acts as a flag for phagocytosis. So if you remember from the steps of phagocytosis video, macrophages, different phagocytic cells would have Fc receptors which allow the easy attachment of the antigen. So again, antibodies, these are involved in the adaptive immune response. We'll talk about them more in chapter 15. But bacteria, in order to be successful pathogens, they need to actually avoid antibodies. So in the chapter 13-14 lecture, this is what we're talking about, avoiding the antibodies so they're not flagged for phagocytosis.